welcome back to Biology B. We're looking at the second lesson of Unit 1. We'll be looking at gene expression overall, but we're going to be focusing on the lac operon. Tomorrow we're going to be doing a lab, and so I would like you to be able to manipulate the lab along with me. So I'm going to ask that you guys download the lab from this website, and I'll have that linked for you in the content. I need you to open the lab. That means you're going to have to enable Flash Player, I think, and play with it. Get in there, make things happen, have a good idea of what I'm going to be expecting of you before we come into class. The same big idea for this unit, and that we're talking about cells being specialized, even though they have the same DNA and genetic code, because they don't always produce the same proteins, causing them to have different functions. So we're going to be looking at a common dysfunction that a lot of us suffer from. The same learning targets: gene expression, make sure you understand what that means and define it. Describe evidence that all body cells contain the exact same genes in the nucleus. Mostly we're going to be looking today at how specialization is the result. And by specialization, I mean we're all a little bit different. In this case, we're going to be focusing on, like I said, a common dysfunction. Let's look at the big picture again. DNA to proteins to gene expression. So on a strand of DNA, you've got versions of a gene that might exist somewhere along this code here, right? And even though everybody has the same genes, they may have different versions that are expressed at different times. So if we're going to look at two different versions of this gene, the normal enzyme would make, we're talking about one that would make a pigment molecule called melatonin, or I should say melanin, not melatonin, that one makes you sleep, melanin in the skin and hair. The defective enzyme would not make this pigment, so this is a defective version. And what you can see when it's expressed is that the normal skin would, in this example, have a lot of melanin, and the albinism version would have not very much melanin at all. They'd have white hair, very pale skin, and eyes, etc. So how is this possible, right? There's a gene that's in your DNA that turns on this melanin, and if that doesn't get turned on, then the melanin's not expressed, and that's how you get albinism. In a nutshell, we're going to talk about some details similar. Differentiation is the name of the game you need to remember. All cells carry full copies of the genome, but it's that different parts of the genome is read and different proteins are expressed, and that's what creates differentiation. All right, so from a stem cell zygote all the way to the formed individual. There are a lot of ways that this process of turning genes on and off could happen. There's a lot of points along the pathway from DNA to a protein that could happen, and even beyond that, you can change the structure of a protein. We'll talk about that in a second. But we're going to be focusing on that happens at transcription. All right, so we're going to be looking at a specific enzyme and how that gets turned on and off when the DNA is transcribed into RNA and mRNA. Like I said earlier, sometimes even after you've made the protein, afterward, the shape of the protein itself can be changed to fit a need. So in the case of insulin, when insulin is made as a protein, it comes with this frame on it, and in order for it to be activated, for the insulin to work, the frame needs to be removed. Insulin, it's the functional version is just this simpler version without that frame that you see around it. So it can happen even after the protein is translated. Also, you need to understand too, one example of gene expression that could result would be cancer. Gene expression can control the amount or ability or likelihood of a cell to divide. So a normal cell dividing would replace tissue that was lost. But if you have a cancer cell situation, the cancer cell does not have the ability to stop dividing. So you end up with a mass of cells where you don't need them. We're again focusing just at the part during transcription. And so while the DNA is read and transcribed into mRNA, transcription factors, this you need to know is a term, are needed to make that happen. And transcription factors are proteins that help in transcription and ultimately to turn the gene on or off. So transcription factors. All right, I've already recorded myself explaining a large portion of this. So what I'm going to do is switch to that video. It's about 5 minutes, and then bring you back just to wrap things up. Let's learn a little bit about lactose intolerance and the genes that we use to control it. The enzyme lactase is not produced in the small intestines of a person who is lactose intolerant. About 50 million Americans have a condition called lactose intolerance. People with this condition can't digest a large sugar molecule called lactose, which is found in milk and other dairy products. The enzyme lactase, which is produced by cells in the small intestine, breaks down lactose in the diet. People who have lactose intolerance stop producing the enzyme early in their life. And so as a result, the lactose sugar molecule moves through the small intestine and into the large intestine, which is where the problem begins. In your large intestines, you have a bacteria called E. coli, and they help you digest everything that you normally eat. But they don't normally digest lactose because it's taken care of earlier in the small intestines. But if a person who has lactose intolerance doesn't digest it in the small intestines, when it comes to the large intestines, those E. coli get a chance to break it down. And when E. coli breaks down lactose, it also produces gas, bloating, abdominal discomfort, and sometimes, unfortunately, diarrhea. So that's lactose intolerance and why it happens. Normally, it's digested into the two sugars, glucose and galactose, in the small intestine. Lactose intolerant people don't do that. When it gets to the large intestine and the bacteria do the job for them, they do it a little bit more messy. E. coli responds to the environment it lives in, or your gut, in order to know whether to express or repress the lactase genes. E. coli prefer to eat glucose because it's the most simple form of sugar, but lactase is a collection of glucose and galactose, so it needs to be broken down before the bacteria can access the glucose. So if the lactose is absent in the environment, your gut, then the bacteria doesn't want to spend energy to make the enzyme to break down something that isn't there. So if lactose is absent, the process won't continue. That's when you'll see a repressor bind we'll discuss later. If lactose is present, it has to break down the lactose to get to the glucose, which is the other half of that molecule. So in that case, the presence of lactose will trigger the bacteria to produce the enzyme in order to break it down. And we're going to discuss those three genes and how this process works here in the next slide. There are 3 genes, each responsible for making one of the enzymes needed to break down lactose. They're located next to each other on the DNA strand. 1, 2, 3 here. Doesn't matter what we call them. The 2 regions on the DNA before those genes are called the operator and the promoter. So those are the 5 key players, plus the RNA polymerase and the repressor that we'll be introducing later. When there's no lactose present, we learned, the bacteria makes a protein called a repressor. The repressor binds to the operator. When the operator is blocked, the genes for lactose digesting enzymes can't be read. In other words, the genes are turned off. So let's see what that looks like. So here you've seen, without any lactose present, the bacteria farther up the DNA strand has a code that codes for this repressor, this blocker of the process, because the bacteria doesn't want to make 3 genes it doesn't need to use. When lactose is present, which you can see here with these green dots, the repressor can't function. We'll show you why that is, but the lactose will bind to it and it won't be able to bind to the DNA strand. With the repressor out of the way, RNA polymerase, which we'll see, will come in and bind to the DNA strand, move along the DNA strand, and transcribe those 3 genes needed to make the protein to enzyme to break down lactose. Here you see the lactose bonding and removing the repressor. In comes the RNA polymerase, which is now able to bind to the DNA strand. Now that the RNA polymerase is able to bind to the DNA strand at the operator and promoter region, it spits out this mRNA message, which is what the instructions will be to code these 3 genes into the enzymes that are responsible for breaking down lactose. We call them lactase. All right, just to recap, the E. coli responds to the gut environment when lactose is present or absent. Again, if lactose is absent, there will not be any transcription. There will not be a protein formed because the bacteria does not need it at that point. It doesn't need to exert the energy to make an enzyme to break down a sugar that isn't there. If lactose is present, everything will proceed as normal. Well, abnormal for lactose intolerant people. All right, just a reminder again, the lac operon is a section of DNA. It has several genes, 3 of them, and a couple of different regions that are the transcription factors. So how is the lac operon expressed and repressed? Remember, it receives signals from the environment. It's like turning off and on a light switch. If lactose is present, then the operon is expressed. If lactose is not present, the operon is repressed. Here's another image of that operon. The players that are missing are the RNA polymerase that would bond to the promoter and slide along it to produce the protein, or mRNA, I should say. Excuse me. The repressor, which would be, if it's present, would bind to the operator and prevent the RNA polymerase from moving forward, much like a speed bump. So, a recap. If the lac operon is blocked or repressed, there's no lactose in the bacterial's environment. The repressor binds to the operator. The repressor blocks the RNA polymerase, cannot bind to the DNA, and transcription will not take place. If the lac operon is turned on or expressed, that's when lactose is found in the environment. The lactose that's in the environment will bind to the repressor. The repressor will change its shape enough to release the DNA molecule. RNA polymerase is able to bind to the promoter, slide past the operator and through those, across those genes that codes for the proteins, which is transcription. So transcription occurs. mRNA is produced. Enzymes to break down lactose is produced. The lactose is further broken down. Alrighty. Okay, so here's some practice for you. I want you to pause the video, screen snip if you'd like to, but I need you to find all of these players somewhere on this picture. So I'm going to pause, and then we'll check your understanding when I come back. Okay, so let's get labeling. The first thing you probably were able to pick out of the whole strand might be this player here. So this player that's going to slide along, here he is again, and the clue that you have for what this player is the fact that this is coming out the back, okay? So this player would be, number 4, would be RNA polymerase. And you know that because you've seen plenty of pictures where a molecule like this is sliding along and producing a tail or a string of molecule at the end. And you should at this point know that number 5 then is the mRNA. All right, that's what's being produced by RNA polymerase. All right, so we've got those two identified. Now, you can look through here and say, all right, operon and operon genes, those look a lot the same. And I already explained to you that the operon is made up of, I'm going to change my color here. It's made up of 1, 2, 3 regions of DNA. They're different genes, plus another area and another area here. So these 5 regions of DNA, this box probably should go like this. So that whole complex would be number 6. That would be the operon. And number 7 is pointing to the genes. So 6 and 7 would be the genes. Now that leaves us with repressor protein operator, repressor gene promoter, and lactose molecule. So if you were to be paying attention to the last part of the video, what you have for 8 is a special segment of the DNA. Let's clean this up. And we'll even zoom in on this. No. Okay. So in this special segment here, you'll see it's got a specific binding site. You see those binding sites there, right? So this section here has a specific binding site that seems to fit this molecule here. So on the DNA strand, there is a segment that a block can fit into, right? So this is a block that can fit, kind of like a speed bump, right? So this would be the operator. Number 8 is the operator. That means number 9, which is farther north, would be the promoter. All right, so that leaves us with number 1, number 2, and number 3. Number 2, if you can notice, is a result of what's happening with this part of the strand above. So that's actually a result of transcription from and translation of this little snip of mRNA, which happens to be from the DNA as well. So this must be another gene. And it's the gene that makes this block, which represses the whole thing. So number 1 must be the repressor gene. And number 2 must be the repressor protein. And that leaves number 3 as the lactose molecule. So I hope you got those right. And if you didn't, at least give yourself a chance here to practice. Let's do some more practice. Some multiple choice questions. When transcription cannot take place, what's happening? So if transcription cannot take place, then the gene is off. What happens when lactose in the environment is broken down? Well, if after all of this process happens in the lac operon, once the lactose goes away again, then there's nothing to bind to the repressor. So the repressor binds to the operator again to block transcription. Letter A would happen when lactose is present. And C is also when lactose is present. All right, A or B, you got to decide which scenario. Number 1, transcription of lactase, that's the enzyme, will occur. Well, the only way lactase will be transcribed is if the RNA polymerase can get to the DNA. So A. Lactose is absent. Well, our intents purposes show that lactose here is the little green circles. So in that case, B. And you don't need the RNA polymerase if lactose is not around. 3. E. coli needs to make lactase. Well, that's when lactose is present, so that's A. The gene will be repressed. That would be B. The gene will be expressed. That would be A. So you guys need to become familiar with this, okay? I'm finally going to leave you with this animated GIF of the Amoeba Sisters video. It encompasses everything. What you can see here at the top is the lactose sugar, which is a disaccharide, binding with the repressor. So the repressor cannot bind to the operator. That allows RNA polymerase to slide past and make that protein. I have another video from Bozeman Science, which is normally 10 minutes, but I've cropped it down to just the lac operon stuff. I will link this into your content as well, and you can access that to take a look. Don't forget that your homework is to download and open and learn how to manipulate the simulation for tomorrow's lab. And it is, okay, so I can give you the link, but it's not going to do you much good. What you want to do is go to the website phetcolorado.edu, and then just type in machine lac operon, or just type in lac operon, okay? So you want to go to the PHET.edu site and search for the lac operon in Biology Sims, and download it, open it, and play with it. Well, I need you guys to check in with yourselves about how you're feeling. Be prepared to come into class tomorrow ready to participate with the lab. And reach out if you have any questions. I'll talk to you later. Bye.